001 block 1 unit 3 we discussed till the point 3.3. Now we are coming to 3.4, that is anterior and lateral temporal lobes and memory. So what is the role of anterior and lateral temporal lobes? So uh, like in earlier studies and whatever we have discussed till now, it has been seen that neocortex is crucial for the storage of memories. So neocortex is important for memories, and then basically storage of memories, we can say. So we can show, if it is involved in that, then we can show like that retrograde amnesia with cortical damage. And even though most amnesias are anterogate means learning new information, problem in learning new information. So most of the amnesias are related to that only. But if neocortex is involved in storage of memories, so whatever we have learned, then uh, if there is a damage in neocortex, right, then there will be retrograde amnesia, because storage information is lost. So that is related to past that you have learned, and that is stored in your long term memory. So we can say, in this way we can say, if there is a damage in neocortex, that can cause a amnesia, right? And then uh, um, a special interest arises in temporal neocortex. Temporal lobe, that I have explained in last class. So temporal neocortex is important for amnesia. So and that is outside the medial temporal lobe. So if any damage happens in lateral cortex of anterior temporal lobe near the anterior pole, lead to dense amnesia that include severe retrograde amnesia. As I said, neocortex is involved in storage of the information, long term storage, you can say. And if there is damage, so we can lose our long term memory, and we call it a retrograde amnesia. That you have learned, whatever you have learned in the past and is stored, that will be lost, and we call it retrograde amnesia. So in such cases, anthrohinal cortex and perihippocampal cortex may be involved. That is one hypothesis that these areas may be involved. So and this retrograde amnesia may involve like decades, like from last 10 years, for you have lost the memory, whatever you have stored from last 10 years, or maybe more, or maybe entire life, whatever you have learned, that is lost if the any kind of you can say damage happens in that area, right? So and there are different problems in that these kind of damages may happen. That one is Alzheimer disease. Alzheimer disease is where like your brain start decaying fast. It is a disease basically related to old age. And there is one more thing, that if something is lost in your brain, like any cells and all, that cannot be regenerated, right? So in Alzheimer is a progressive disease. There is like you can slow the speed of that, but you cannot uh, stop that thing. So gradually this area also get affected, and it may lead to retrograde amnesia. And similarly in different infections it happens, and one of the infection is herpes simplex. That is herpes simplex and Alzheimer. lobes, right? And that can cause such condition like retrograde amnesia. Then dense retrograde amnesia might still form a new memories. And even if there is a retrograde amnesia, people can form new memories, right? Dense means like a severe kind of amnesia. And this type of amnesia is called isolated retrograde amnesia. Isolated retrograde means you have lost your previous memories, but you can form new memories also at the same time. So it is particularly related to damage of anterior temporal lobe, and this position of the temporal lobe is not therefore essential for acquiring new information. So uh, so suppose you have damage in anterior temporal lobe, then you can lose your previous information, we call it memory, and but you can create a new memory at the same time. So there is no interrogate amnesia, only retrograde amnesia is there. And so we can say in this condition, if we can say like temporal lobe is not essential for acquiring new information because there is a damage and still you are able to learn new memories. So you will the temporal lobe is not involved in acquiring new information. So this was like the role of lateral temporal, anterior and lateral temporal lobe. Recent studies neuroimaging studies. Neuroimaging studies are where we see the activity of brain using MRI, magnetic resonance imaging, or PET, positron emission tomography. Uh, so in that, uh, studies, like we have found that memories are stored in different parts of neurocortex, neocortex, and where like these are like encoded and represented, and where we associate information from other information. So in neuroimaging studies indicated that our you can say these information that we have stored or distributed across neocortex, right? So that is why when some area if damage is there, though still we have some memories. So that is what it is saying, right? So now we will discuss animal models of memory. So animal models of memory is where you study animals and you do experiment on animals on different brain areas. Sometimes you remove that part or you stimulate that part and you study how it affects the humans, animals, and on that basis you can predict the role of brain in human, because we have restriction on conducting researches or removing any part of the brain of human beings, right? So that we will discuss in 3.5 topic, animal model of memory. And basically these studies have been done on monkeys, है ना? And with lesions in hippocampus and surrounding cortex, and that gives us you can say invaluable information about temporal lobe and memories. So and these animals, basically monkeys, they are more closer to human beings. That is why we take these animals, right? And in this study the question is, if we say the question is how much the hippocampus alone as compared with surrounding structure in the medial temporal lobe, participated in memory deficit, like patient like HM that we have discussed earlier. So how this hippocampal alone is involved in our memory. So till now, like we have understood that hippocampus and amygdala and other things are involved in memory storage. And other thing is like if you will like observe this unit, so everywhere there is one name that is common is temporal lobe. So in memory, temporal lobe is very important, right, that we have to understand. And amygdala is also important, hippocampus is also important. So when the hippocampus and amygdala both were removed surgically, uh, the Miskin 1970 has of like NIMH found that the amount of impairment as a measured on test varied according what was lesioned. Means where it was there, where was like lesion, it depends on that. Okay, if we remove amygdala and hippocampus, so what we have removed, how much we have removed, that is directly related to the loss in memory on measured test when they have measured that memory on that test, right? And then Messikin found that in monkeys, the memory was impaired only if the lesion uh, included the hippocampus and amygdala. So when hippocampus amygdala is lesion mean when there is like some like you have created removed that thing. And this led to an idea that amygdala is a key structure in the memory, and this idea does not fit with the data from the RB. RB that we have discussed is a case of HM and RB that we have discussed in last chapter. And as RB had Retrograde Amnesia, where he lost like the ability to acquire new knowledge, and caused by a lesion restricted to the neurons of the hippocampus and no damage to amygdala. So when the study was done on monkeys, hippocampus and amygdala both were like removed, and and it was found that monkey has some memory loss. But in RB's case, there was an retrograde amnesia, and the lesion was restricted only to the neurons of hippocampus, and no damage was done to amygdala, right? So so it was not going with that study. Then Stuart, Jola and colleagues of University of California, they have investigated this thing. So they performed more selective lesions of the brains of monkeys, and distinguishing between amygdala and hippocampus as well, and as the surrounding cortex near each other. So what they have done is they selectively removed hippocampus. They selectively removed hippocampus and amygdala, and they surgically created lesions. Surgically created lesions means they have done surgery to create lesions. You can say in Hindi like a kind of like wound and something, so created. And the surgically created lesions of the amygdala and ethorhinal cortex, or the surrounding neocortex of the parahippocampal gyrus and like peripheral cortex. So and in their study, it was found that you can say the lesions, let me remove it. The lesions of the hippocampus and amygdala produces the most severe memory deficit only when the cortex surrounding these region was also lesioned. So suppose there is a this is amygdala and this is hippocampus and this is the surrounding area. So they found that ki if only hippocampus and amygdala is removed, and along with in along with this, if we remove other parts nearby, then it is more severe form of problem they have created in memory. So when the lesion of the hippocampus and amygdala are made but the surrounding cortex was spared, only these two parts, amygdala is removed, not the surrounding parts. The presence and the presence or absence of the amygdala lesions did not affect the monkey's memory, and they found कि only these parts are removed then, and with no damage in the amygdala area, then जो monkeys का memory था that was less affected, है ना, or it was not at all affected. So the amygdala when could not be part of the system of the support acquisition of nerves. So when lesion in amygdala was not related, it was found that the amygdala was not related to that kind of memory loss. So the amygdala then could not be part of the system supported the acquisition of long-term memory. So it is not related. So it is proved by this study that amygdala is not related to acquisition of long-term memory, acquisition and learning of long-term memory. And then Jola and his colleagues selectively created lesions in surrounding cortex of perihinal and entorhinal and parahippocampal regions. So then what happened is this worsened the memory performance in delayed non-matching to sample test. So it worsened the memory performance, है ना. So and in follow-up work, when they have followed up that thing, that lesion of only the parahippocampal and perihinal cortex also produce significant memory deficit. So if we created lesions in parahippocampal and perihinal cortex also produce a significant memory deficit. So if we have damaged those areas, then also it is significant memory deficit in them. So next study, if we say what we have found in that study, the parahippocampal and perihinal areas receive informations from the visual auditory somatosensory association cortex and send these inputs to hippocampus, from there to other cortical regions. Means parahippocampal and perihinal areas receive information from visual, auditory and somatosensory association cortex. There are three kind of you can understand in this way. There is three kind of cortex माने area जो है. So one is where the information is received, right, and other is where that is associated with other information. Suppose you have seen some food, right, and it is looking very, you can say, tasty to you, and there is a information that is coming from your nostrils, that is smell. So when they are associated with each other, then you can make a good decision that food is very nice, right? So that is what these somatosensory, when you touch it, something like that. So these two areas actually, they receive information from visual, auditory and somatosensory association cortex. They take information from there, whatever is sensitized after getting the information from the environment, and they send this information to the hippocampus, where they have sent they send it to hippocampus. And from there to other cortical regions, and from there it goes to other cortical regions, right? And this hippocampus cannot function properly if these vital connections are damaged. Suppose these two areas, like hippocampal area 1 and then this other area, these are the two areas, and they are getting information from a different association cortex, and different association cortex, and this is the connection. If we cut that in connection, and this information goes through neurons, you can say that, you know. So if we cut this information, and then this information will not reach to parahippocampal and perihinal areas, when it will not reach there, and this connection is damaged, so what will happen? This will not, because it is not getting information, so it will not work properly, though there is no damage there, only the connection is damaged. And then so there will be some memory loss, because you are not able to make connections, right? So whatever information you are getting from outside. But more than this, we also know that these regions are involved in much processing themselves, and hence lesion restricted to the hippocampus do not produce the severe form amnesia, and do lesion include surrounding cortex. So what happens is, understand in this way, these like hippocampal areas also like process information. So when we cut this information, this information reaching to this hippocampal areas, then it is not much, amnesia is not that much severe, like memory loss is not that much severe, right? And then if we delete, like remove or lesion this, including surrounding areas, so this includes these connections also. So when that is damaged, then what happens? This and this, like this one and this one is damaged, then there is a severe memory loss. So that may be a cause of severe memory loss when we remove surrounding area also. So that is what this study says. I hope this will be clear to you, right? Then if we talk in like about all this in summary in animal studies, so we can say, let me clean it first. Okay, in summary, the data from animal are highly consistent with the evidence from amnesic patient like RB and HME that we have discussed earlier. That implicates that hippocampal system in the medial temporal lobe, the associated cortex, as a critical for forming long-term memory. So surrounding area as well as the hippocampal area both are important in forming long-term memories. So if any lesion that damage the hippocampus directly or damage the input आउटपुट रिलेशनशिप ऑफ हिप्पोकैम्पस विद नियोकॉर्टेक्स, यानी जो इंफॉर्मेशन आ जा रही है, produce severe memory impairment. That is why it creates severe memory impairment that we have discussed. And the amygdala is not crucial part of the system for episodic memory, but it is important for emotional memory. Understand one thing, there may be a question कि amygdala is like for your you can say emotional memories which involve emotions, right? So amygdala is not crucial for system, इस system का part नहीं है, but the episodic memory, when you remember about any episode, like when like you have celebrated first episode, that is a part of long term memory actually, so that is affected. But when it comes to emotional memory, when you get so emotional memory, you attach some emotional meaning to that memory, so that is affected if amygdala is lesioned. So understand that thing. So moreover, the animal data match well with those from amnesic with regard to perseveration of short-term memory process after medial temporal lobe has been damaged. So monkey's memory deficit in the delay mismatching to sample task make more pronounced that interval between the sample and the test increase. The medial temporal lobe, then it is not essential for short-term memory in working memory process. So we have discussed what is working memory. So the conclusion is कि all the animal researches, like, they indicated the same thing as we have done researches on humans, like RBs and HM cases. So this was the conclusion, you can say, related to these studies, and how it affects. So to summarize, there is like hippocampus and amygdala. Hippocampus is involved in memory, long-term memory, and that and amygdala is related to emotional memory. And if we damage hippocampus and amygdala, or we don't create any lesion in amygdala, so hippocampal only like create memory loss, but that is not that much serious. But we remove if we remove the surrounding parts also, then that creates a more severe memory loss. So it indicates that the connection between like other parts of the brain, that is neocortex of different areas, is damaged, then there is a severe memory loss. So that is what animal studies indicated, right? Okay, so now we will discuss the imaging the human brain and memory. So in earlier days when we were studying, uh, you can say, our brain, so we were dependent on people like whom are like who are like have damaged brain due to some accident, tumor, or any kind of infection and extra. But with the development of the technology, what happened is, we have advanced imaging imaging systems like fMRI, like functional magnetic resonance imaging, PET, positron emission tomography. So that kind of things are there where you can see the brain functioning while the person is doing some task of like learning or memory or any other cognitive functions. So that created a huge difference, in a sense that now you can study normal human being also. You can study when they are working, they are learning something or some material, and then at the same time you can say how the brain is functioning, which part of the brain is active and all, right? So that was the like very effective way to work on human brain without damaging anything, right? So we are talking about memory from last 2 3 lectures, right? So but in this chapter, nothing is given about types of memory and everything, so I thought, okay, I will add that thing in this chapter. So now we can study the brain in a different manner. So in this context, like in this topic, we are going to review the studies related to this episodic memory and semantic memory, procedural memory, and perceptual representation system. So first thing first, like we should understand what are these memory types, because until unless we won't be able to understand what it is, because we are discussing these terms from last 2 lectures, I think 1 lecture. So we have to understand these kind of memories. So all of them are like long term memories. So I have this thing, like you can check, that is our memory is like can be divided into one and two parts. That is short term memory, we have already discussed. इसका जो स्टोरेज होता है 5 ± 2 जो स्टडी हुआ था प्रीवियस लेक्चर में हमने डिस्कस किया. Then long term memory, and long term memory is divided into two parts, that is explicit and implicit, right? Explicit is that is conscious, that you use, you can retrieve, that kind of thing. Unconscious, that you are not aware about, that is working on their own, right? So like we will study that, like we will see different researches related to episodic. So episodic is like events happen to you, like at when you have passed like your 10th exam, or when was your 16th birthday, or when like you moved to some other place, when you joined the job. That is episodic one. That is episodic, right? These kind of things we call episode, because these are the episode in your life. And then semantic memory, you can say semantic memory like general knowledge of word, like whatever we are saying, like it is written like memory. So initially when we have learned this language, so we learn like what is M, what is E, M, and what is O, R, or Y. What does the different alphabet we have learned, then we learned how to associate them, how to make the word out of those like alphabets, and how we pronounce it, what is the meaning of that. So all things are related to semantic memory, right? Then implicit, as I said, it is unconscious, like priming means like if I say black, tell me the opposite of the black, so you immediately you will say white. And when I say like height, high, you will say low. So this is the priming. Once the word is coming, opposite word, you will immediately tell. So these kind of things are like priming, you are primed for that, right? And then we are coming to procedural memory, like motor, like initially when you learn driving, like maybe car or bike, so you have to focus lot of attention on those things. But after some time, you are listening song, but you at the same time you are driving also. If someone comes in front of you, you accordingly you can change the like speed of your car or bike. So that after learning that skills, motor skills, you are like that goes into unconscious, and it works there immediately. It works, well, there is no need to focus on that. So then you can do one two thing at a time, like that is like while driving, like you are talking on phone, though it is dangerous, because you may like miss the information that is coming to you from the road, because accordingly you have to respond. So that is not advisable. But driving car become unconscious process. So that is what procedural memory. So this is and that is why we are saying that we are going to study episodic memory, semantic memory, and procedural memory. That is the motor one, how you do anything, like that is this one implicit memory, procedural one, right? So that, and then another system is perceptual representation system, PRS. The stages are related to that also. So that also I have included. Perceptual representation processes, like whatever you have learned and how you recognize immediately something, that is what perceptual memory, perceptual representation system. So it is a memory system whose purpose is to recognize item and terms, and permitting rapid and identification of formerly encountered stimulants. Like you have information about this, in this example you have information about chair, what is chair, and as soon as you have seen, you see the chair, you immediately recognize. You don't have to be like very much like aware about those things, what it is, how it is, that is, but so you know that it has like four legs, some back, and then place for sitting it. So and then like whenever, immediately as you get that information from the environment, you see it and you immediately you recognize it. So we call it perceptual representation system. So the purpose of discussing these things, so that you can understand what are the studies like we are going to cover and what they are doing. So two things understand, like in this like like we have imaging systems that can be like researches can be done on normal people also, not only the people who have damaged brain, right, or animal studies. So now I am coming to the episodic and encoding and retrieval. So given a purported role of hippocampus, like we have on a hippocampus system in encoding memory long-term storage, researchers have eagerly addressed the issue using positron, jo PET I have said, like told you. So how it works? So they used PET, positron emission tomography, right, and the functional magnetic resonance fMRI. What two things I have told you. So so in that we can see which part is active when we are encoding or recognizing some information. So now as we know that hippocampus, like we have studies in damaged brain, so the question is was whether the hippocampus because active becomes active on encoding new information. So now because of these techniques we can see if someone is learning or encoding, so is these or these part are active during that period of time. So they're like these people, James Maxwell, Leslie, uh, Ungerleider and their colleagues 1996 at the NIMH National Center of Mental Health presented subject with the picture of their faces and nonsense pattern and using PET. Okay, they have given some nonsense pattern and faces. So nonsense patterns means जिसका कोई meaning नहीं है, and faces like that you can recognize. And the task is to remember a face and recognize the face and perceptually analyze the face, comparing two faces. All those tasks were included in that research. And when they were doing this, PET, any positron emission tomography, was done to see the neurological activity, cerebral blood in cerebral blood flow. So whenever your any part of your brain is active, so there lot of like brain sorry, blood flow increases in that area. So that is was it was intended to identify, right. So when that one task is given to them of encoding and storage, and then we are using PET to identify those hippocampal areas or any other areas which is active during that task. So what they have found, they have observed that right hippocampus region was activated during encoding. So right, as I told you, there are two hemispheres in brain, right and left. So right, so every they are symmetrical. I know. So so right hemisphere means the part of the hemisphere that that is like sorry, right hippocampus means the hippocampus part that is situated in the right hemisphere. Okay? During encoding, encoding of the face, but not during the recognition. So when we were encoding the face, yani we are remembering the face, like encoding, like storing the face, then right hippocampal was activated. And when we were like recognizing it, recognizing means when we see and find out who is this or what face is this, and that is recognition. So it was not active, right. So while retrieval process should have been engaged, and these data are consistent with those amnesic patients who had medial temporal lobe damage that led to the interrogate amnesia but preserved distant retrograde memories. Like right hippocampus area was activated, and during the recognition it was not activated, है ना, where the retrieval process was engaged. Retrieval means when you are taking out some information from your memory. So what they have found, it was consistent with the previous studies, brain damage studies. These are the imaging studies, and that were brain damage studies. So it was consistent with that, right? So why it was it like it were these were like consistent with that, because same finding we have found on brain damage studies, right? Encoding also activated the left prefrontal lobe. So encoding yani when we are learning that information and storing it, where recognition activated the right prefrontal. So encoding is related to left prefrontal area, and recognition is related to left prefrontal area. These two things, right? Encoding is related to left, and recognition is related to right, right? So these things were there. So we have more support of hippocampus role in memory, and now we have, because we have done neuroimaging on normal individuals who don't have any kind of damage and all. So now we have more like studies that is confirming that hippocampal area, yani hippocampus, is like has an important role in memory, है ना? And as well as like hemispheric, like support for hemispheric asymmetries in memory function. Hemispheric means like this is one part right, left, and this is right, suppose. So left and right, so there is role of left and right areas also, that we call hemispheric asymmetries. Asymmetry means this work, this like left is doing something and right is doing something else. Like one is involved in encoding, one is involved in retrieving or recall of the memory. So that is what we got. So encoding and retrieval process were lateralized. Lateralized means right and left part क्या क्या करता है, that is lateralization, and respectively giving rise to model with acronym of HERA. HERA means they have created this acronym, that is Hemispheric Encoding Retrieval Asymmetry. So what is the asymmetry? Symmetry means दोनों एक तरीके से हैं, but asymmetry means कि this is doing something else, this is doing something else. So that is asymmetry. So we call it HERA. HERA is like hemispheric encoding retrieval asymmetry. यानी कि we got to know about asymmetric role of these things. This model represents the idea that encoding involves the left hemisphere more than the right, and retrieval involves the right hemisphere more than the left, and both processes predominantly involve the dorsolateral prefrontal cortex. Which area? Dorsolateral prefrontal cortex. And in encoding and retrieval information from the long-term memory, neocortical areas were the most activated. Which areas? Neocortical areas that we have found in like animal studies also. So that is like we should know. Some more studies at MIT, Harvard University, and other universities, and they found that like event related responses were larger in prefrontal and medial temporal lobe. Event related if something is happening, any event is happening. And during encoding of the words and pictures, when we are talking about word and pictures, then that were later remembered. So when like there is something related to event, then prefrontal and medial temporal region, and during encoding of words and pictures. So if we are encoding words and pictures, so that part is activated. So that is all about like these researches. And now we are coming to semantic encoding and retrieval. Semantic, you know that semantic is like when you process the words and all, that is semantic one. So the encoding and retrieval of semantic knowledge, and when you are learning words and grammar rules and everything, also have been studies using the functional neuroimaging. So that also we use functional neuroimaging. Now you know that fMRI and PET scans, etc. So significant new findings have been uncovered. What we have found, in a particular evidence that for domain specific organizations like knowledge of animate and inanimate objects is localized in different cortical regions. Like when we are again some animate, like who are like living beings and who are not living beings, so they're different cortical regions means your brain areas are activated. So it shows that we are like storing the information category wise, like has proved to be fascinating story. And unlike episodic retrieval that activates the right prefrontal cortex, semantic retrieval involves the left prefrontal cortex. So when we were talking about episodic one in previous topic, so it was right prefrontal lobe which was activated. In semantic it is left prefrontal. So again this is a same hemisphere difference, any asymmetry. So, and the reason includes Broca's area. Broca's area, Broadman's area 44 extending into area 46. Broca's area is a part which is very important in producing word. Motor, in a motor sense. Motor sense, when I am talking, how my tongue will move, how my lips will move, how my like you can say whatever movement is required for the for producing the word is required, that is controlled by Broca's area. And Broadman's area 44 and 46, it is like it is kind of division that is done by Broadman's की different areas का number उन्होंने दिया है, and that is what Broadman's area is. So that Broadman area is 44 and 46. And the interventral lateral region, ventral lateral region, Broadman's area 44 and 45. This these were that were activated, and the lateralization to the left hemisphere remains regardless of whether the memory is being retrieved or of object or word. So lateralization, yani difference between both the hemisphere is like remained there instead of like there is no actually effect of either you are retrieving the objects or you are retrieving the words. So that we have found in this semantic encoding and retrieval studies, right? Now we are coming to procedural memories. Like now you know what is procedural memories. Procedural memory, when you learn some procedure like driving a car or something, doing something in a different, like in a stepwise manner, motor thing when we talk about. And uh, and this is like implicit memory, that you have learned that motor sequence, how you drive cycle, how you drive a motor car, that kind of thing, that is procedural and knowledge, right? So in this study what we found is like there are like these people like Scott, Grafman, Elliot, Hayes, Hazeltine, and Ivory in 1950 investigated the brain basis of procedural motor learning in normal subjects, normal who doesn't have any kind of brain dysfunction. So he tried to study that how the procedural learning is there, what happens in the brain when someone learns kind of things, right? They compared the condition in which subject learn motor sequence implicitly during dual task condition, which help to prevent subject from explicitly noticing the learning the sequence. And like in dual task, you are doing one thing and as well as the other thing, like you are driving a car, you are also talking on phone, that kind of thing, like you are doing two things at a one thing, and how you learn those things. So PT conducted during the dual task condition demonstrated activation of the motor cortex. Cortex is a part of the brain that is involved in motor learning and motor related activities, and supplementary motor area of the left hemisphere. So left hemisphere motor area left hemisphere का activate होता है. So in general, like if I'll tell you, so each co- lobe is divided into three parts, as I said earlier, like sensory, who collect the sensory information, then motor, and then association cortex, where we associate all the information. So motor cortex is there. That is why I told you motor cortex and the supplementary motor areas of the left hemisphere, and the Putman's in the basal ganglia bilaterally. Bilaterally means both side. As I said, they are symmetrical. So bilaterally they are activated. Also activated were the rostral, prefrontal cortex, and parietal cortex. And therefore, when the subject were implicitly learning, any unconsciously learning the task, brain areas that control limb movement were activated. So when you are like doing one motor task and doing something else, so limb movement, like for movement, your limb movement is required. So and unconsciously that area that controls your limb movement was also activated. When the distracting auditory task was removed, when we have removed the auditory task, like I said, when you are talking on phone as well as you are driving, the right dorsolateral prefrontal cortex, right premotor cortex, right putman, and parieto-occipital cortex were activated bilaterally. When you have removed that was, then these were activated bilaterally, दोनों side में activated थे. So this was the study ki how they which part is active when you are involved in dual task, when you are involved in only one task. So that is what in your examination, if you will write, I'm not saying you write exactly what it is, but if you will write ki this part is involved in this, these kind of studies have been done, and then you will get good marks. You can get good marks, and that will increase your understanding of the subject also. So now the next topic is perceptual priming and implicit and explicit memory. Perceptual priming, that we have discussed, like perceptual recognizing, how you recognize anything, as that chair wala example that I have given to you. So there are people that they have studies this kind of thing. In that, Daniel Saster and his colleague of Harvard University, they studied the neural basis of perceptual priming, implicit learning in PET study. So perceptual priming, how you recognize immediately anything, that we have discussed in that example of chair. So that they were, because that is very important task, because you cannot as and when you see anything, you just recognize it. So the scanning was performed only during the task. So during the task we have done the scanning, they have done it. Subject manifested implicit priming behaviorally. Priming मतलब immediately होना कोई चीज. No activation or deactivation were noted in the hippocampus. So it was done immediately, and there is no activation of like hippocampus, but blood flow in the bilateral occipital cortex decreased, and the hippocampus was not activated then, even though implicit perceptual learning was obtained. So hippocampus was not activated. And bilateral जो आपका occipital cortex था, है ना, was like it was like said that no activation was deactivation were noted in the hippocampus, but blood flow in the bilateral cortex decreases. So bilateral cortex of occipital cortex. Occipital cortex is related to your perception. your information, like whatever information sensorily, like from eyes you are getting. So वहाँ पे वो decrease हो गया था, though when you are seeing something it must have increased. So so you can say the in priming, yani perceptual priming, hippocampus was not activated, and your occipital area में जो blood flow था that was decreased. So you can say hippocampus was not involved in that activity, perceptual priming. The conclusion from this and other studies that implicit and explicit retrieval information is subserved by separate brain system. So this is by separate brain system, like in like अगर आप कहें other type of memories, hippocampus was active, but in this hippocampus was not active. So that was we found in perceptual priming, implicit and explicit memory. So there is a difference between implicit and explicit memory when you in terms of activation of brain areas. So in all these history, what we have found, you can say conclusion is that the hippocampus encodes new information but also retrieves recent information when explicit recollection is involved. Perhaps more interestingly, deactivation of the visual cortex for previously seen words in is a correlate of perceptual priming. So when you are saying something, you are seeing something, and your visual area, that is occipital cortex, is not that much activated, there is a low blood flow in that area, so it indicates that it is not a burden on your occipital system, because that information you already have, only you have to match that what it is to perceive that object. So it indicates that there is priming, immediately you are recognizing something. So in this way, the neuroimaging studies, they have identified the neural activation of different areas and in a consistent way, and how these human and animal models are like work, and we can strengthen our like view on that, and through these studies, that whatever we have found in lesion studies, any brain damage studies, and neuroimaging, they were matching to each other. Okay, so now we are going to discuss cellular basis of learning and memory. So understand what is cellular basis of learning. So cellular basis of learning is you can say cellular basis of learning जो होता है, that is kind of, because if you have learned about brain, so brain we call brain cells neurons, like brain cells or neurons. So and these are like neurons, that structure I have made, this is neurons. So this is pre synapses, and ये है post synaptic, and this is synaptic cleft. So they are not connected, the neurons, like they communicate each other with each other. So brain process the information through neurons. Suppose you have seen something, that will go to your brain, and then brain process the information chemically actually. So there is some chemicals, we call it neurotransmitters, that comes here, and then here, and there are some gates, they open, and these gates also open, and that bind that neurotransmitter to this neurons, and that goes to other neuron, the information goes to other neuron, that is the neural processing, you can say. So what happens is and there is a relationship that strengthen in certain cases when in cases where we learn, and there was and that is what like cellular basis of memory and learning. So there was a person, we call them Hebb, right? Donald Hebb, in 1949, right? So he told us about these things, and we call it Hebb's law. We call it Hebb's law. So Donald Hebb in 1949, Hebb's rule state that if a synapse between two neurons, this is a synapse, two neurons repeatedly activated about the same time the hyposynaptic neuron fires, structure the chemistry of the neuron changes, and the synapse will strengthen. This is known as Hebbian learning. So means repeatedly you are getting some information. So what happens is there is like this relation between this and this neuron is strengthened, and when it is strengthened, that is learning. So because they learn a way ki how to fire, how and what kind of things are there. So this relationship is strengthened, and we call it Hebbian learning. When maybe you can understand in like very simple, when you are doing something again and again and again, so same process is chemical process is happening in your brain, and that's in strengthening one day by day, and it creates like learning. So that is Hebb's law. So a more general and more complex, and if there is a more general process or complex process, mechanism is called long-term potentiation. Is and so we call it long-term potentiation if more complex process is happening there or more general process is happening there. In this process, neural circuit, in this process long term potential, what is long term potential? Long term potentiation means strong हो रहा है relationship, long term potentiation का मतलब ये हुआ. तो in this process neural circuits in hippocampus that are subjected to repeated and intense electrical stimulus develop a hippocampal cells that becomes more sensitive to stimuli. So these cells are in hippocampal area also, because everything is made up of neurons, because these are the basic cells of the brain. So in hippocampal area, right, and during the process of LTP, long term potentiation, there is a like intense electrical stimulation, and then and they develop a hippocampal cells that become more sensitive to stimuli, in this particular stimuli. So when it becomes more sensitive to it can easily be activated when this kind of information is getting to you. So maybe that is what is required in brain for proper learning, or you can say the priming and all that happens. So that is what that happens at cellular level. That is why we call it cellular basis of learning. So long term potential, so these are the like few neurotransmitters like glutamate, NMDA, these also, so that you can read because that become more complex. So I'm not going into that. I'm just telling you what it is and how it affects. So long-term potential and memory performance, the effect of the enhanced response can last for weeks or even longer, suggesting to many that this could be a mechanism for long-term memory. So what happens there in the cellular at cellular level during the process of long-term potentiation, and that can last for weeks, even more longer, and that may be a cause of long-term memory that we have to understand. And if in any ways we disrupt this process, if we disrupt this process, then this learning may be hampered, or we can disturb the learning. So that is what cellular basis of learning. And this is it in this unit 3. So in this we have learned what are the different areas involved in learning and memory, what are the like cellular processes involved in learning and memory, and what are the like areas involved we have seen through fMRI and PET scans. So all these things we have learned in this unit.